Hi, my name is Audrey Braun, and I am a graduate student at the University of California, Riverside, and my research focuses on contaminant uptake in produce, specifically related to the combined use of recycled irrigation water and biosolids in agricultural systems. So let's get into it. Today, I'm going to present on the land application of biosolids and the bioaccumulation of contaminants in fruits and the estimated human dietary intake. So biosolids are nutrient-rich byproducts from wastewater treatment plants, and about 10 million tons are produced globally each year. And one sustainable way that we manage this material is through land application, which improves soil and plant health while avoiding detrimental disposal methods such as landfilling and incineration. However, previous studies have shown that biosolids can also contain a wide range of organic contaminants, and some of these may accumulate in food crops, raising concerns about human exposure. As for knowledge gaps, most studies on contaminant uptake have been conducted under controlled conditions and focus on vegetables or feed crops. And importantly, there are no studies examining biosolids applications to fruit trees and the potential implications for human exposure. So addressing these gaps is critical for identifying safe and sustainable biosolids for use. To address this, I designed a field study focused on fruit trees grown under realistic agricultural conditions. And the objective is to evaluate uptake of biosolids-borne contaminants into fruit and assess potential human exposure through consumption. So based on prior work that I've done, showing how contaminant levels in showing that there are low contaminant levels in vegetables, I hypothesized that fruit trees with their longer growing periods and larger biomass will accumulate even fewer contaminants in edible tissues. So ultimately, this work aims to support the safer use of biosolids in fruit production. This large-scale field study includes apple, avocado, mandarin, and peach trees fertilized with biosolids at agronomically realistic nitrogen-based rates. So I'm going to be using 4 biosolids treatments, and the fruit will be analyzed for 24 contaminants, including PPCPs, industrial additives, flame retardants, and PFAS compounds. So the fruit, soil, and roots were collected at harvest, and it will be analyzed using a catcher-spaced extraction method followed by LCMS. And then human dietary exposure will be estimated using US EPA consumption data. As for the proposed timeline, the trees were grown to fruiting maturity, treated with biosolids, and harvested over the past year. And then sample extractions began in October, but after a temporary instrument outage, our LCMS analysis is starting up again. And I expect to complete analysis and data interpretation in the coming months with manuscript submission targeted for June. And then my results will be shared through a mix of written, oral, and collaborative outreach. A manuscript will be published in a peer-reviewed journal and shared through reports developed with my partners that include the Water Research Foundation and the US EPA. And then in May, I will be participating in a roundtable discussion with these collaborators, as well as other academic and industry partners. And then I want to present this work at local, national, and international conferences that include CTAC and Canvas. And then I believe that together these efforts will help ensure that the research is communicated to scientists, regulators, and stakeholders involved in biosolids and agricultural management. So yeah, thank you for listening to my research proposal, and please reach out to me if you have any questions. Thank you.